Dear students, this is Dr. Pooja Thakkar, a biology expert, and we are in the fifth session of cell biology. Here we need to discuss regarding mitochondria. As you can see in the diagram, mitochondria is basically a double membrane structure. The outermost membrane is very basic and very common, but the inner membrane is folded into many segments, as you can see here, right? So those kinds of segments are called cristae. Now the cristae are mainly responsible for increasing the surface area of the membrane so that it can do more and more respiration. Now how they'll do the process of respiration, that we will understand in the next slide. The other labels are given here is the ribosomes, which are green in color, which are typically present in the mitochondria, as well as the purple colored structure which is called DNA. So mitochondria is having its own DNA as well as ribosomes so that they can make their own mitochondrial proteins. Now those kind of DNA as well as ribosomes are typically present in a fluid which is called matrix, which is there into the lumen. That lumen is formed from the internal membrane compartments. Fine. Now you can also see the dotted structure over here on the cristae. Those kind of structures are basically called ATP synthase, or they can be called ATPase, which are basically having a head and a tail like structure. The F0 portion is called tail, whereas the F1 portion is called the head. And these structures are crucial for the formation of ATP inside the matrix of mitochondria. Now in this particular upcoming slide, we will discuss regarding mitochondria, which was primarily or preliminarily discovered by Colliger. They are stained by Michaelis using the particular stain called Janus green. So they'll stain blue or green colored particles in the colorless cytoplasm so that you can study it easily. They are double membrane bound structure, as we have discussed, the outer membrane and the inner membrane, and they are dividing its lumen into two compartments. Now one thing I've forgotten, and that was intermembrane space. That intermembrane space is the space between the two membranes, that is outer membrane as well as the inner membrane. Fine. Now what the particular outer membrane is doing, which is basically a permeable membrane which will allow the metabolites to come in or go out by the presence of the specialized proteins called porine proteins, which are forming channels for the transportation of molecules across the membrane. But the inner membrane forms a number of infoldings called cristae, as we discussed, towards the matrix. These cristae are basically tubular in the plant cell, as well as they are plate-like in the animal cell as well as in the fungi cells. These inner membrane bears oxysomes or ATP synthase, which are responsible for the ATP synthesis process, or they'll work as ATP synthesis machine. And those machines are connected with the electron transport chain. That electron transport chain is basically a structure where varieties of electron carriers are there. We'll discuss this kind of electron transport chain as well as oxysomes function in detail during the 14th chapter of respiration. Okay, and these all systems are basically involved in the ATP synthesis, and so mitochondria are considered as the powerhouse of the cell, as they are mainly responsible for the respiration. Now the matrix of mitochondria, which is the fluid inside, which contains circular DNA molecule as well as few RNA molecules, ribosomes, as well as the machinery which is mainly responsible for the protein synthesis. Now these kind of mitochondria are completely absent in the anaerobic cells, as they are not at all doing any kind of respiration. They are dependent on the others and the RBCs. Now RBCs are basically produced in bone marrow, so they do not need to undergo any kind of multiplication, as well as they are only having the energy to perform the oxygen transport or the carbon dioxide transport, that is the gaseous exchange process. So they do not need extra energy, so they are definitely not at all containing any mitochondria. These mitochondria are in the other sense, you can say, semi-autonomous organism, and they multiply by the division process. Now what do you mean by semi-autonomous here? Autonomous is they are doing their own task, by their selves. Semi-autonomous, they are slightly dependent on the cytoplasm for certain action. Otherwise, in the evolution, we can say mitochondria were initially working as the aerobic bacteria. So they are having their own genome, that is the DNA or the RNA as well as the ribosome, to make their own mitochondrial protein, and they are basically responsible for the survival of those kind of aerobic bacteria in the previous atmosphere. But occasionally what happened during the course of evolution, these kind of aerobic bacteria get engulfed, or they got entered into the developed eukaryotic cell, and then they are called mitochondria, and they became the permanent part of the highly developed plant cell as well as the animal cell. So, what are the functions of mitochondria? So, as we discussed, they are mainly involved in the production of energy. The simpler nutrient molecules are to be sent into the mitochondria, the basic units. They are processed inside and they produced charged molecules inside. जैसे कि for example, the glucose is sent in, that get broken down, and they'll form the charged molecules which are combining with the oxygen molecule to form the ATP. So this is the overall process that we will discuss in the chapter 14, as I told, which is called oxidative phosphorylation process. The second important task is they are helping the cells to maintain the proper concentration of calcium in different compartments of the cell. As we know, calcium is playing a very significant role in various tasks of the body. Building certain molecules, mitochondria are responsible for building certain components or molecules of blood, as well as the varieties of hormones, that is testosterone as well as estrogen. Mitochondria, especially of liver cells, have the enzymes which are detoxifying the ammonia, as well as, in other sense, you can say they are mainly involved in the detoxification process of the body, right? Here I have given the basic structure, or the morphology, which is morphological features, or you can say the structures which are basically present in the inner mitochondrial membrane, like complex 1, which is the NADH dehydrogenase, the complex 2, which is fumarate reductase, complex 3, which is mainly involving cytochrome complex 4 is also cytochrome oxidase complex, and the complex 5, as we discussed, here you will be able to see the ATP synthase or ATPase enzyme. So these kind of assembly of 4 complexes with the 5th complex, that is ATPase, is present in the inner mitochondrial membrane, and they are doing the molecule breakdown process to form the charged molecules. These kind of charged molecules will produce as electrons. The electrons are transferred from one complex to the another complex by using electron carriers, and those electrons will be finally transported to the complex 5, which is ATP synthase. And at the same time, proton gradient is also maintained by this particular complex on which sides, towards the inner side, that is the metric side, as well as towards the intermembrane space, which is the membrane which is the space between the two membranes. Here you will not get the idea in detail, but these things we'll discuss later. So this is the overall process which is performed by mitochondria to produce ATP. So this is all about mitochondria. This is very easy to understand. You just need to mainly focus on the diagram here and the components which are present in them, so that you will get the idea in detail when we'll discuss the physiology context of this particular chapter regarding the context to mitochondria. Okay, so thank you so much for this. Kindly do like the video, understand it very carefully. You can ask the questions in the comment box below, and please subscribe the channel, that is Inside Biology. Thank you so much.